Chapter 22. Restoring Signaling Precision. Signaling precision depends on the ability of the membrane to present a consistent environment in which receptors detect input, assemble into functional complexes, and terminate activity within defined time frames. Precision is not created at the level of the signal itself. It emerges from the structural conditions that determine how that signal is interpreted. When membrane composition varies, identical inputs produce different outputs across cells and tissues. Restoring precision, therefore, requires rebuilding a membrane environment that enforces uniform rules for activation, amplification, and termination. Receptor activation depends on controlled proximity. Ligand binding increases the likelihood that receptors interact with one another and initiate signaling, but this process requires a balance between mobility and constraint. If the membrane is too rigid, receptors cannot move into position efficiently, reducing activation. If too fluid, receptors encounter one another randomly and outside of organized domains, producing unstable or incomplete signals. A membrane composed of stable lipids maintains a range of fluidity where receptors can cluster within defined regions, allowing activation to occur only when input reaches a sufficient threshold. Microdomain organization governs where signaling occurs. Specific lipids and proteins segregate into regions that concentrate receptors and downstream components. These domains provide a controlled environment for signal propagation, ensuring that interactions occur in a coordinated sequence. When membrane structure is inconsistent, these domains become unstable and signaling components disperse. This disrupts the sequence of interactions required for precise output. As composition stabilizes, domains reform and signaling pathways regain spatial organization. Signal amplification depends on the integrity of these domains. Once activated, receptors recruit additional proteins to propagate the signal. This process requires stable anchoring points within the membrane. When the surrounding lipid environment supports consistent packing, these interactions proceed efficiently, producing a response that reflects the original input. In unstable membranes, amplification becomes uneven, with some signals expanding excessively and others failing to propagate. Termination defines the boundary of signaling events. Precision requires that signals end once their function is complete. This involves dissociation of receptor complexes, deactivation of downstream components, and restoration of baseline conditions. Membrane structure influences how quickly and reliably these processes occur. Increased permeability and disorganization prolong interactions and delay termination, leading to overlapping signals. Restoring structural integrity allows signaling complexes to disassemble in a controlled manner, re-establishing clear boundaries between events. Crosstalk between pathways is reduced as precision improves. In unstable membranes, overlapping signals interfere with one another because components are not confined to specific regions. This creates noise within the system, where unrelated pathways influence each other. Organized domains limit these interactions, allowing pathways to operate independently while still integrating at higher levels when required. Energy availability supports signaling precision. Activation, propagation, and termination all require ADP. When mitochondrial efficiency improves and gradients are maintained, energy supply becomes consistent, allowing signaling processes to proceed without interruption. This reduces variability in response and supports the timing required for coordinated function. System-wide coordination emerges from local precision. When individual cells interpret signals consistently, tissues respond in a unified manner. Hormonal inputs produce aligned effects, neural signals propagate with predictable timing, and immune responses remain proportional to input. This alignment depends on the convergence of membrane properties across tissues, which is achieved through sustained remodeling under stable conditions. Restoring signaling precision, therefore, involves rebuilding the structural environment that governs how signals are processed. By establishing membranes that support controlled receptor movement, organized domains, and reliable termination, the system regains the ability to translate input into proportional and coordinated output. The improvement is not driven by stronger signals, but by a framework that ensures signals are interpreted accurately at every stage. Re-establishing receptor environments. Receptor function is determined less by the presence of the receptor and more by the membrane environment that surrounds it. Every receptor operates within a localized lipid field that defines its mobility, orientation, and ability to interact with other proteins. When this environment becomes inconsistent, the receptor remains intact but loses the conditions required to translate binding into a controlled signal. Re-establishing receptor environments means restoring the physical constraints that allow receptors to activate, propagate signals, and disengage in a predictable manner. The first requirement is controlled lateral movement. Receptors must be able to move within the plane of the membrane to encounter other receptors and signaling partners, but that movement must occur within defined limits. A membrane composed of stable lipids provides resistance that prevents random diffusion while still allowing directed interactions. This balance ensures that clustering occurs only when ligand binding increases the probability of interaction, preserving threshold-dependent activation. Local packing density determines receptor conformation. Many receptors undergo structural changes when activated, and these changes depend on the mechanical properties of the surrounding lipids. When packing is too loose, the receptor lacks the resistance needed to stabilize its active configuration. When too tight, conformational transitions are restricted. Reintroducing a lipid environment with consistent packing allows receptors to shift between states in response to input rather than being constrained or destabilized by the membrane itself. Microdomain formation provides spatial organization for signaling. Specific lipid compositions create regions where receptors and downstream components concentrate. These domains act as platforms that bring together the elements required for signal propagation. When membrane composition supports domain stability, receptors localize to these regions, increasing the efficiency and specificity of their interactions. Without this organization, signaling components remain dispersed, and the sequence of interactions required for precise output is disrupted. Electrical conditions within the membrane influence receptor sensitivity. Ion gradients and local charge distribution affect how receptors respond to ligands and how downstream signaling pathways are initiated. Increased permeability alters these conditions, reducing the consistency of receptor activation. Restoring membrane integrity stabilizes these gradients, providing a uniform electrical environment that supports predictable receptor behavior. Protein-lipid interactions define receptor anchoring. Many receptors interact directly with specific lipids that stabilize their position and orientation within the membrane. When these lipids are replaced by structures that do not support these interactions, receptors become less stable and more prone to irregular movement. Reintroducing lipids that support these interactions restores anchoring, allowing receptors to maintain proper alignment with signaling partners. Termination of signaling depends on the ability to separate receptor complexes after activation. This requires a membrane environment that allows controlled disassembly without prolonging interactions. When structure is restored, receptors disengage from one another and from associated proteins in a timely manner, preventing overlap between signaling events. Distribution of receptors across tissues becomes more consistent as environments stabilize. Differences in membrane composition between cell types decrease, allowing receptors to respond similarly to the same input. This reduces variability in system-wide responses and improves coordination between tissues. Energy availability supports all aspects of receptor function. ADP-dependent processes maintain gradients, facilitate protein interactions, and drive the recycling of receptors after activation. When mitochondrial efficiency improves, these processes operate without interruption, reinforcing the stability of receptor environments. Re-establishing receptor environments, therefore, involves reconstructing the lipid context in which receptors operate. By restoring controlled mobility, consistent packing, organized domains, and stable electrical conditions, the membrane provides a framework required for receptors to function with precision. The result is not an increase in receptor number or signal strength, but a restoration of the conditions that allow existing receptors to produce accurate and proportional responses to input. Normalizing hormone response. Hormonal signaling depends on the ability of tissues to interpret circulating signals in a consistent and proportional manner. Hormones are distributed system-wide, but their effects are determined locally at the membrane, where receptors detect and translate those signals into intracellular actions. When membrane structure varies across tissues, the same hormone concentration produces different responses, creating a mismatch between signal and outcome. Normalizing hormone response requires restoring a membrane environment that allows receptors to operate under uniform conditions, so that input is translated into output with consistent magnitude and timing. Receptor sensitivity is defined by the physical properties of the surrounding lipids. Hormone receptors rely on controlled fluidity and stable packing to transition between inactive and active states. When membranes become too permeable or disorganized, these transitions lose precision. Some receptors fail to activate efficiently, while others remain active longer than intended. Re-establishing a balanced lipid environment restores the mechanical constraints needed for receptors to respond proportionally to hormone binding, aligning activation with signal strength. Signal propagation depends on the assembly of downstream components within organized membrane regions. Hormone binding initiates a sequence of interactions that amplify the signal and produce a cellular response. This sequence requires spatial organization so that proteins encounter each other in a defined order. When membrane domains are unstable, these interactions become inconsistent, and the signal either weakens or spreads beyond its intended scope. As membrane composition stabilizes, these domains reform, allowing signaling pathways to proceed with predictable efficiency. Termination of hormonal signaling defines the boundary of response. Hormone action must cease once the required effect is achieved to prevent overlap with subsequent signals. Membrane structure influences how quickly receptor complexes disassemble and how effectively downstream pathways return to baseline. Increased permeability and disorganization prolong signaling, creating a state where responses overlap and interfere with one another. Restoring structural integrity allows signals to terminate within defined time frames, preventing accumulation of activity. Transport of hormones and secondary messengers is also affected by membrane conditions. Many signaling molecules must cross membranes or move within them to reach their targets. When permeability is altered, this movement becomes less controlled, affecting both the speed and distribution of signaling components. Stabilizing membrane structure ensures that transport occurs through regulated pathways rather than through uncontrolled diffusion, preserving the specificity of hormonal effects. Energy availability supports the entire signaling process. Hormone response requires ADP for receptor recycling, phosphorylation events, and maintenance of ion gradients. When mitochondrial membranes regain the ability to contain proton gradients, ADP production stabilizes, providing a consistent energy supply for signaling. This reduces variability in response and supports the timing required for coordinated hormone action. Intertissue coordination improves as responses normalize. When membranes across different tissues converge towards similar structural properties, receptors interpret signals in a more uniform manner. Hormones that regulate metabolism, growth, and stress produce aligned effects, allowing the system to respond as a cohesive unit rather than as a collection of independently behaving compartments. Feedback systems regain effectiveness under these conditions. Hormonal regulation relies on feedback loops that adjust signal output based on response. When responses are inconsistent, feedback signals become distorted, leading to over- or undercorrection. As receptor environments stabilize, feedback loops operate with greater accuracy, maintaining hormone levels within ranges that produce appropriate responses. Normalizing hormone response, therefore, involves restoring the structural conditions that govern how signals are interpreted at the membrane level. By rebuilding a consistent lipid environment, receptors regain the ability to activate, propagate, and terminate signals in alignment with input. The system does not require stronger signals, but a stable framework that allows existing signals to produce accurate and coordinated effects across tissues. Stabilizing inflammatory signaling. Inflammatory signaling depends on controlled initiation, spatial confinement, and timely termination. These conditions are set at the membrane, where substrate availability determines which mediators can be produced, and where receptor organization determines how signals are amplified and resolved. When membrane composition becomes inconsistent, both the generation and interpretation of inflammatory signals drift. Stabilization requires re-establishing a substrate environment and structural context that limit unnecessary initiation and support completion once signaling has begun. Initiation is governed by the availability of fatty acid precursors embedded within the membrane. Enzymatic release of these substrates produces signaling molecules that drive inflammation. When the membrane is enriched with highly reactive and easily mobilized lipids, the threshold for initiation decreases, allowing signaling to begin with minimal stimulus. As composition shifts toward more stable lipids, the rate of spontaneous or low-threshold activation declines. This does not eliminate inflammatory capacity. It restores the requirement for a defined trigger before signaling begins. Amplification depends on receptor clustering within organized domains. Once mediators are produced, they bind to receptors that initiate cascades affecting neighboring cells. This process requires spatial coordination so that signals are contained and directed. In unstable membranes, receptors are dispersed or form transient clusters that amplify signals unevenly. Some regions overrespond, while others fail to propagate the signal. Rebuilding membrane organization allows receptors to localize within defined regions, producing amplification that reflects the original input rather than random variability. Termination is the critical step that defines whether inflammation resolves or persists. Resolution pathways depend on the generation of mediators that actively suppress signaling and restore baseline conditions. These pathways require specific substrates and a membrane environment that supports their production. When substrate availability is limited or membrane structure disrupts enzymatic activity, termination signals are insufficient. The response continues beyond its intended duration, creating a state of persistent activation. Stabilization involves ensuring that both the substrates and structural conditions required for resolution are present, allowing signaling to complete its cycle. Membrane permeability influences the spread of inflammatory signals. Increased permeability allows mediators and ions to move beyond their intended range, expanding the area of activation. This reduces the ability to localize responses and increases the likelihood of system-wide effects from a localized trigger. As membrane integrity improves, diffusion becomes more controlled and signaling remains confined to appropriate regions. Oxidative processes interact with inflammatory signaling at multiple levels. Reactive species generated during inflammation can further damage membrane lipids, increasing susceptibility to additional signaling events. This creates a feedback loop where inflammation promotes conditions that sustain itself. Reducing oxidative load and stabilizing membrane composition interrupts this cycle, limiting the propagation of damage and allowing resolution mechanisms to function effectively. Energy availability supports both activation and termination. Inflammatory signaling requires ADP for mediator synthesis, receptor activity, and cellular responses. Resolution processes also depend on energy to restore baseline conditions and repair damaged structures. When mitochondrial efficiency improves, the system can complete the full signaling cycle rather than remaining in a prolonged activated state due to insufficient energy for termination. Intercellular coordination improves as signaling stabilizes. Immune cells communicate through mediators that depend on consistent interpretation across tissues. When membrane conditions align, these signals produce coordinated responses that are proportional and localized. Variability decreases, and the immune system operates with greater precision. Stabilizing inflammatory signaling, therefore, involves controlling both the substrates that initiate the response and the structural conditions that govern its progression. By restoring membrane composition and organization, the system regains the ability to initiate inflammation when required, amplify it within defined boundaries, and terminate it once its function is complete. The result is a signaling process that operates within controlled limits rather than persisting as an unresolved and self-reinforcing state. Improving system coherence. System coherence emerges when tissues interpret inputs under the same structural conditions and produce aligned outputs over time. It is not created by increasing signal strength or adding regulatory layers. It depends on restoring a shared membrane baseline so that transport, signaling, and energy production follow the same rules across organs. When membranes differ in composition and behavior, each tissue processes identical inputs in a different way, and coordination breaks down. Improving coherence requires reducing that variability until responses converge. Signal interpretation becomes uniform as receptor environments stabilize. Hormones, neurotransmitters, and local mediators arrive with defined concentration and timing, but their effects depend on receptor behavior within each tissue. When membrane composition converges, receptor clustering, activation thresholds, and termination kinetics align. The same input produces a similar response profile across cells, reducing conflicting outputs. This allows endocrine and neural signals to coordinate rather than compete. Transport regains consistency when permeability and gradient control are restored. Substrate movement depends on maintained differences in concentration and charge. In unstable membranes, passive leakage competes with regulated transport, creating uneven distribution. As membranes rebuild with tighter packing and controlled fluidity, gradients hold with less energy, and transporters operate under predictable conditions. Nutrients and ions are delivered according to demand rather than drifting toward regions of least resistance. Energy distribution stabilizes as mitochondrial membranes regain containment. Efficient proton gradients increase ADP yield per unit of substrate, reducing the need for compensatory overconsumption. When energy production becomes consistent across tissues, processes that depend on ADP operate with less variability. This supports synchronized function, as cells no longer experience alternating states of surplus and deficit that distort signaling and transport. Temporal alignment improves when activation and termination occur within defined windows. Many physiological processes depend on timing, including hormone pulses, neural firing patterns, and immune responses. Membrane instability prolongs or shortens these windows unpredictably. As structural integrity returns, signals begin and end within consistent durations, allowing processes to sequence correctly rather than overlap or interfere with one another. Feedback systems regain accuracy under coherent conditions. Regulatory loops depend on consistent relationships between input and output. When responses vary between tissues, feedback signals misrepresent the state of the system, leading to overcorrection or persistent imbalance. With aligned membrane behavior, outputs reflect inputs more reliably, and feedback loops adjust signals with greater precision. Intertissue communication becomes more effective as variability decreases. Signals transmitted through blood or neural pathways are interpreted similarly across different organs, allowing coordinated adjustments to changing conditions. The system operates as an integrated network rather than as independent compartments responding in isolation. Remodeling cycles reinforce coherence when the substrate pool supports stable composition. Each cycle reduces variability by replacing divergent structures with a common set of lipid properties. Over time, differences between tissues narrow, and the system approaches a unified baseline. This does not eliminate specialization, but it ensures that specialized functions operate within a consistent structural framework. Energy demand for maintenance decreases as coherence improves. Fewer resources are required to correct imbalances caused by inconsistent signaling or transport. This allows the system to allocate energy toward productive processes rather than continuous compensation, further stabilizing function. Improving system coherence, therefore, involves converging membrane properties across tissues so that inputs are processed under shared conditions. As variability in structure declines, signaling, transport, and energy production align, allowing the body to function as a coordinated system with predictable and efficient responses to internal and external demands.